Welcome to the Deep Dive. We're here to take complex academic papers, pull out the really key insights, and talk them through, giving you a solid understanding without you having to wade through the dense text yourself. Today, we're looking at something quite fascinating. It's a paper that sits right at the intersection of, well, citizen science, cutting-edge tech, and marine conservation. It's from Frontiers in Marine Science, published just this year, 2024. Lead author Frederick Leerhøi. The title is Exploring the Potential of Extreme Citizen Science with Danish High School Students Using Environmental DNA for Marine Monitoring.
Yeah, and that word extreme is doing a lot of work there. This isn't just about, getting kids interested in science. It's about showing that you can actually decentralize some pretty advanced molecular ecology. They basically took an existing citizen science project and, pushed it further. They empowered high school students to become genuine collaborators, co-creators, really. These students are gathering data, really critical data, on Danish marine life using eDNA. And the kicker is, this data is intended to be good enough to potentially inform government conservation policy.
Okay, hold on. That sounds ambitious. High school students running molecular labs and producing data reliable enough for policy. Let's unpack that. What exactly is this eDNA tool that supposedly makes this possible?
Right. So eDNA, environmental DNA, it's essentially the genetic material that organisms leave behind in their environment. Think skin cells, mucus, waste products, all floating around in the water. Instead of trying to physically find and count, say, an elusive eel or track an invasive jellyfish, you just take a water sample. Then you analyze that water for the specific DNA signature of the species you're interested in.
Okay, I get the concept, but why is that the key for this kind of large-scale citizen science?
Well, it has a couple of big advantages in this context. First, it's non-invasive. You're not disturbing the animals. And crucially, the students doing the collecting don't need to be expert taxonomists. They don't need to visually identify maybe a tricky fish species or tiny plankton. They just need to follow a very clear protocol for collecting the water and filtering it through a small cartridge. It's called a Sterivex filter.
Ah, okay. Simpler fieldwork.
Exactly. And because the collection is relatively straightforward, you can get massive geographical coverage. You could have hundreds of students sampling all along the coastline at roughly the same time. That's perfect for national surveillance.
And this wasn't a brand new idea for them, right? This extreme version built on something already running.
Yes,
Yeah.
absolutely. It grew out of a project called DNA in Life, which is based at the Natural History Museum of Denmark. That project started way back in 2014, and it already involved, I think, over 10,000 students.
Wow. Okay, so they had a big pool of participants already engaged.
They did, and that initial groundwork was vital. In the, let's call it the original DNA in Life phase, the students did the field work, collecting the samples, doing the filtration out in the field. That's more like traditional citizen science. But the really complex lab work, the DNA extraction and the PCR analysis, that was mostly done back at the museum by the professional academic staff.
So the students collected, but the pros analyzed.
Pretty much. Students might visit the museum lab sometimes to maybe run a final test under supervision, but the critical sensitive steps, those were kept centralized.
Still, even that sounds like a great educational experience. Getting hands-on with sample collection and then seeing the high-tech analysis.
Definitely. The educational value was always high. They were seeing real advanced molecular biology in action applied to environmental questions. It makes science feel relevant,
Okay, so now let's get to the extreme part, the big shift that started around 2022. What changed?
This is where they really upped the ante. They made the decision to essentially trust the students and their teachers with the most technically demanding parts of the process, the DNA extraction and the PCR itself.
How did they manage that logistically? Sending samples back and forth still takes time.
Right. And time is critical for preserving DNA quality, especially eDNA, which can degrade. So to shorten that distance and time lag, they did something pretty bold. They set up actual functioning DNA labs inside two of the participating high schools, Schering and Herning gymnasiums.
Full labs in high schools. That's quite a commitment.
It really is, logistically and scientifically. Because now, the students and teachers at those schools are doing the entire workflow, from sample to result. The teacher effectively acts as the lab manager. That's true, extreme citizen science.
Okay, but I have to ask the obvious question. Contamination. Molecular biology, especially anything involving PCR, is incredibly sensitive to it. A tiny stray molecule can ruin everything. How on earth did they trust high school students and, frankly, high school environments with this?
That is the critical question, and the paper tackles it head on. The answer boils down to incredibly rigorous, standardized quality control, QC. They knew everything depended on it.
So what did that look like in practice?
Well, first, the teachers. They were identified as the crucial gatekeepers. They had to complete a mandatory, intensive one-day training course, not just on teaching science, but specifically on performing these advanced molecular procedures correctly and safely. Then, everything was standardized, detailed protocols, checklists, and importantly, video guides for all the technical steps. Even things like how to handle a micropipette accurately, which honestly takes practice even for pros. Or properly cleaning lab benches.
Micropipetting is definitely a skill. And qPCR, the analysis method they used, that needs precision too.
Absolutely. qPCR, quantitative real-time PCR, it amplifies tiny amounts of target DNA so you can detect it, but will also amplify any tiny bit of contamination. That's the big risk. A false positive could make it seem like a species is present when it's not.
So how do they stop stray DNA floating around a potentially busy school lab from causing chaos?
Multi-layered approach. Physically, they mandated strict cleaning routines using specific chemicals, 5% chlorine, then 70% ethanol on all work surfaces before and after use. Chemically, they used pre-aliquoted reagents. That means students never handle the main stock solutions. They get small, single-use amounts. That drastically reduces the chance of contaminating the main supplies.
Smart. Minimizing handling errors.
And logistically, even in maybe small lab spaces, they insisted on separating the area for DNA extraction from the area where the PCR was set up,
Right.
with a full clean down in between. And maybe most importantly, one eDNA sample processed per lab per day. That's it. Slows things down, maybe, but massively reduces the risk of sample-to-sample contamination. That focus makes the data trustworthy.
Okay, so strict protocols, but how did they check if it was actually working? How did they validate the results coming out of these school labs?
Through the qPCR results themselves. Every single run, every plate of samples included controls. A positive control containing known target DNA had to amplify properly, usually before a certain cycle number, like cycle 41 in the study. That proves the test chemicals and machine are working. And critically, a negative control, usually just sterile water, had to show absolutely no amplification signal. Zero. If the negative control lit up, it meant contamination, and that entire batch of results was thrown out. No questions asked.
So only results where the positive control worked and the negative control is clean were considered valid.
Exactly. They also used a conservative cutoff point for detection, ignoring very faint signals appearing late in the run after cycle 41. That helps avoid interpreting potential background noise as a true positive. High confidence was the goal.
And the result of all this effort, let's talk about the data they actually generated. The scale seems impressive.
It really is. The paper summarizes findings from samples collected between 2017 and 2023. We're talking about 3,300 students and 140 teachers involved. And they covered most of the Danish coastline and fjord systems. That kind of spatial coverage is incredibly valuable for environmental management. It's something a small research team could probably never achieve on their own.
Okay, so what did they find? The paper focused on 5 key species, right?
Yes, 5 species chosen for their ecological or economic importance in Danish waters.
Let's start with the sea walnut, Nemiopsis lady. That's an invasive jellyfish, isn't it?
It is, yes, an invasive ctenophore, technically. The eDNA monitoring gave them a really clear picture of its seasonal patterns. They consistently found the highest detection rates between September and November.
Which means coastal managers can target their monitoring or mitigation efforts much more effectively during that peak season.
Precisely. Then there's the European eel, Anguilla anguilla. This one's critically endangered, so getting accurate distribution data is vital.
What did the eDNA show for the eel?
Well, encouragingly, they found its DNA pretty widespread. It was detected in more than half of all the sites sampled, across both open marine areas and important fjord systems, which are nursery grounds.
That kind of geographical map is gold dust for conservation planning, right? Helping decide where to focus protection efforts.
Absolutely. Essential data for red-listed species. Next up, the round goby, Neogobius melanostomus, another invasive species, this one a fish.
And what was the story there?
This was actually one of the more surprising results scientifically. They detected the goby's eDNA in some northern regions of Denmark, where it wasn't previously thought to be well established.
So the students basically provided an early warning signal.
Kind of, yeah. It challenges existing assumptions about its distribution limits and gives authorities a heads up. It allows them to potentially act before the species takes a major hold in those new areas. That's a huge advantage of this kind of widespread monitoring.
That really highlights the power of the approach. What about the other 2 species?
We also produce some interesting ecological insights. European perch, Perca fluviatilis, traditionally thought of as mainly a freshwater fish. But the eDNA detected it quite often in open, brackish waters, particularly in eastern Denmark. This suggests it might be more adaptable to salty conditions than previously assumed. Good context for understanding its potential range.
And the last one, sea fire. Sounds ominous.
It can be. Alexandrium austenfeldi. It's a type of microscopic algae that can produce toxins and cause harmful algal blooms, which can impact shellfish safety and public health.
So monitoring it is important for safety.
Very much so. The eDNA data confirmed it has a broad distribution across Danish waters. Again, valuable information for public health agencies and fisheries managers who need to know where the risk areas might be.
Okay, so stepping back from the specific species, what's the really big takeaway here? What does this study fundamentally demonstrate?
I think the core message is proof of concept. It shows convincingly that high school students and their teachers, if they have the right training, rigorous protocols, and support, can generate high-quality eDNA data. Data that's potentially good enough to support serious scientific research and even inform national environmental policy. The rigor is absolutely the key.
It feels like a successful example of, well, decentralizing science and technology transfer, taking sophisticated tools out of the university lab and making them work effectively in a much wider network.
Exactly. And the practical implications are significant. The insights they gained, especially on the endangered eel and those invasive species, provide the kind of spatial and temporal data that really helps make evidence-based decisions about managing coastlines and ecosystems.
But we should also be clear about what this specific setup can't do, right? The limitations. You mentioned the qPCR setup was geared towards screening.
Yes, that's a really important technical point. Because they optimize for educational use and high-throughput screening, basically asking, is the DNA present, yes or no? They didn't include standard dilution series in their qPCR runs.
What are needed for?
For accurate quantification. Professional labs use those dilutions to determine precisely how much DNA is present, down to a defined limit of detection, LOD, and limit of quantification, LOQ. This student setup is fantastic as an early detection tool finding where a species is. But it can't reliably tell you the concentration of DNA, which often correlates, though imperfectly, with the abundance of the organism.
So it finds the needle in the haystack, but doesn't tell you how big the needle is.
That's a good analogy, yeah. It's a common aspect of many eDNA studies, but particularly relevant here, where quantification steps were simplified for the citizen science context. Another general limitation, of course, is that detecting DNA doesn't always mean a live organism is right there at that moment. DNA can persist for a while.
Understood. Looking forward, what are the hurdles to scaling this up even further, making it even more widespread?
Interestingly, the main challenge they highlighted wasn't really about the student's ability or the cost or the logistics of the labs. Those seem solvable. It was more about the teachers.
Oh.
Even with the training and the free local lab access, some teachers apparently still found the advanced molecular methods a bit too complex or maybe intimidating to implement consistently in their teaching schedules.
So it's more of a confidence or comfort barrier with the technology for some educators.
It seems that way. Overcoming that is likely key to expanding the network further. It's an intellectual or pedagogical hurdle rather than a purely technical one.
And the immediate next step for the research team, what do they need to prove next?
The big goal now for this extreme DNA in life phase is to collect enough data to do a direct, robust comparison. They need to show statistically that the results generated in the high school labs are just as reliable and consistent as the results generated for the same samples run in the main controlled laboratory back at the Natural History Museum.
Proving that data equivalency.
Exactly.
Yeah.
Once they nail that comparison, the model is essentially validated. Then they can confidently push for wider adoption, maybe include more schools, and definitely expand the list of target species being monitored.
It really is a fascinating project. It pushes the boundaries of what we typically think citizen science can achieve. It's not just data collection, it's complex analysis directly contributing to national monitoring.
It empowers students, makes their learning incredibly relevant, and potentially provides a cost-effective way to gather crucial environmental data on a massive scale.
So perhaps a final thought for you, the listener, to consider. We've seen how this Danish project used high school students and eDNA under strict quality control to monitor marine species invasives, endangered ones, even harmful algae.
Yeah, the question it raises is quite powerful. If this model works for marine monitoring in Denmark, what other really complex national-scale scientific monitoring challenges could we tackle using this kind of distributed, rigorously controlled, extreme citizen science network? Think about air quality, maybe tracking agricultural pests, or even public health surveillance like monitoring wastewater for disease markers. Could students be the key to gathering that data too? That's the provocative potential here.